Today we're going to talk about medical school histology basics. We'll be covering the accessory glands of the digestive system, cover the liver, the gallbladder, salivary glands, and the pancreas. Talk a little bit about the duct system they're in, the cells that are there, the origin of the cells. Thank you. Medical school histology basic: liver, gallbladder, salivary glands, and pancreas. Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about the accessory organs for the digestive tract. And here we can see in this Body Worlds rendition of a female and male, you can see the diaphragm here, and there's the liver, the pancreas in through there, the stomach would be in there as well, duodenum, liver, gallbladder. We can see the different organs associated with the digestive tract. Our objectives are to understand the general organization of these accessory organs of the digestive system and how they contribute to obtain metabolites necessary for growth and energy for the body. We want to learn the origin of these glands and how these structural features of these glands contribute to their function of digestion and absorption of food stuff. And here we can see the organs that we'll talk about. The liver, and you can see the classical lobule of the liver, the gallbladder, which is associated with the liver on the side, salivary glands located at the top, and the pancreas. Those are the ones that we're going to study. Start out with, we'll have the salivary glands, liver, gallbladder, pancreas that we'll talk about today. Now, the function of the digestive tract is to move food, secretion of digestive juices, and absorption of foodstuff, and the accessory glands contribute. Salivary glands lubricates things for things to slip down. Also, the salivary gland and the pancreas secrete digestive juices, as well as the liver secretes a bile for emulsifying fats and getting rid of cholesterol. Liver stores nutrients and it cleans the blood. It also is associated with digestive organs that contribute antibodies and antibiotic viral growth substances to prevent infection, as the digestive tract has to maintain a milieu in the body that is different as pure a state as possible. Now, the origin of the digestive tract is the endoderm. You've got the digestive tube, as well as the liver, pancreas, gastric glands, and intestinal glands. They come from the endoderm. And here we can see the endoderm coming through, giving rise to the lungs, as well as it gives rise to the stomach, and a outgrowth of the gallbladder, and even the liver, and the pancreas as well. So you can say that most of the organs associated with the GI tract, as well as the GI tract, come from endoderm, and you can see how one pipe gives rise to the other organs. And so we have the salivary glands, we have the liver, the gallbladder, and the pancreas that we'll be talking about today. One of the most important glands, of course, is the liver. It filters blood. It has a lot of Kupffer cells, which are macrophage type cells. They're monocyte origin of the cells, and they filter the blood. There's also stored blood in the sinuses. It maintains normal glucose concentrations by regulating uptake of glucose, or the breakdown of glycogen and glucose maintains a certain normal level to help maintain homeostasis and metabolism and transport of lipids. And it secretes plasma proteins, clotting factors it does, and nutritional metabolism of bile, drug metabolism, drug tolerance. The smoother you are can clear the drugs. If you're giving someone like anticonvulsant drugs, for example, you have to keep giving a patient more and more of them because the liver clears them. Also, it produces bilirubin, and bilirubin in your blood makes you jaundice, and it secretes bile for emulsifying fat. So the liver does a host of different things, very important for its contribution to the digestive tract. And here we can see this is a classical lobule. We can see it here in a pig. The pig is good because it's got connective tissue around the classical lobule, and a classical lobule has a central vein. So blood is coming in from these portal radicals and through this area and percolates down into the central vein that we can see. And so here we can see on a pig liver, we can see the classical lobules, one right here, one right there, and it has a central vein in the center, and it's separated by connective tissue you can see in between. So this is a classical lobule, not a classical lobule. And also the liver has a capsule. Sometimes it's fairly thin, sometimes it's fairly thick. But in either case, it has a mesothelium lining the cavity. Here we can see some of the cells. These are hepatocytes, this monkey liver, and these are Kupffer cells that have phagocytizing. Here we see blood cells. Now the liver has a couple of functions. It acts like an endocrine organ. Also, it acts like an exocrine organ, and you can see it secretes glucose, plasma proteins directly into the blood like an endocrine organ, but it also secretes bilirubin as an exocrine organ as well. This dual role of these is important for compartmentalization of these endocrine exocrine-like products. The liver has to have certain architectural makeup, and hepatocytes is arranged in a fenestrated. You can see a column through here, anastomosing and anamosing columns of one thick layer with a sinus on either side. And so you can have two sinuses, and also you can have the bile canaliculi in between. Here we can see the bile canaliculi in yellow, and then those drain into a bile duct that you can see here in green, and then you see the cords of cells, and you can see the sinuses that runs between the cords of cells. So the hepatocyte has to have contact to the sinuses, as well as a way to keep the bile separated from the blood. And here you can see the portal radical. You have portal vein coming from this. This is your hepatic vein coming in from the small intestines. There's a bile duct, and there's hepatic artery bringing oxygenated blood in through here. So the blood comes from here, percolates down through there by these hepatocytes, and then finally out through the central vein. So you may have different surfaces of the hepatocyte. You do. Here you can see one to interact with a blood sinus on one side, and an interact with a blood sinus on the other side as well. In addition to that, it has a bile canaliculi on either side. So this one has four surfaces, two for exocrine, and then two for endocrine as well. And here we can see the hepatocyte has lots of mitochondria through the rough ER, smooth ER, glycogen stored. But here you can see the two surfaces. This is a sinusoid where the blood's up through there, and then this is where the bile is discharged in the exocrine function. So the liver is the largest gland, and you can see the hepatocytes in through there where the sinus is going down in between there. You can see the holes in the sinuses, and then it goes into the central vein. So it produces bile salts. Also, the endocrine function, it produces albumin. Albumin is very important for osmotic pressure of the blood, and also fibrinogen, clotting factors, things like that are produced directly in the blood as endocrine. So here we can see the makeup of part of a classical lobule with the central vein in the middle. And this is your triad. There's three parts to it. You got the branch of the hepatic artery bringing oxygenated blood that mixes with the portal blood that's unoxygenated because it already come through the small intestine, and then that percolates through here, and nutrients are then stored in the hepatocytes, and also toxins are detoxified in this process. So the blood goes from these portal radicals to the central vein. However, the bile is in the bile canaliculi, and that goes back toward the portal radical, opposite from the direction of the blood, and it is in a bile duct for it to be discharged in the stored in the gallbladder and then discharged in the small intestines. And here we can see the portal radical, the bile duct. You can see the cuboidal cells in through there making up that duct, artery, and the hepatic vein, and you can see cords of cells in through these. These are hepatocytes. This is the blood sinuses that are on either side of those cells. You can see there are individual cells lined up, touching sinuses on either side, and also the bile canaliculi is in between to two different cells. Here we see the classical lobule again, with connective tissue surrounding, separating those, and you can see the three components of the portal radical, which is located here and here and here, all around, and all of these contribute to blood in the central vein. So if you see one of those as portal radical, you can see the bile duct, hepatic vein, hepatic artery here, and that's the same as here. And then blood would be percolating down through here, ultimately going to the central vein that we're seeing right here. And these are the hepatocytes that we see. Another specimen, we can see the same thing, the bile duct, the artery, the hepatic vein, which is a part of the portal system, hepatic portal system. You can see the cords of cells located here and the central vein. If we look at the different cells that are there, the main cell, the main parenchymal cell, is the hepatocyte. You can see its nucleus, and there's the limits of the cell with lots of mitochondria in there that we can see as the dots that we can see there in this electron micrograph. You can see sinuses in through there, and also you can see the little spots, little white spots here. That's the bile canaliculi. And so we can see hepatocytes in different places. Also, we see Kupffer cells here. They have taken up carbon, so they're phagocytic cells that line the sinuses too. You can see one here, use it across the sinuses, as you see there, and then we can see where they've picked up debris in this electron micrographs. So these are cells that are derived from blood monocytes, and they are macrophage-like cells, which are part of the lining of these blood vessels. And then you have endothelial cells. Those endothelial cells are the additional cells that line the sinuses that are there. You have Kupffer cells and the endothelial cells lining the sinuses. And here you can see them all together. The endothelial cells, the Kupffer cells, and the hepatocyte. If we look at the hepatocyte here, we can see big mitochondria through there. Also, we'll see a bile canaliculi, which has tight junctions on either side to keep the bile separated from the rest of the from the blood. And here we see lots of glycogen stored in the liver, lots of rough endoplasmic reticulum for making proteins. Also, some Golgi apparatuses. Here we see a gap junction between adjacent cells that we can see. Lipid that's there. Also, there's a fair amount of smooth endoplasmic reticulum. I can't see it right now. Nucleus up here. And the smooth endoplasmic reticulum is associated with detoxification. Here we can see one of those sinuses in through there, the hepatic sinus, and the endothelial cells that line that lumen. Just inside we have the space of Dease. This is a space of Dease here where microvilli project up from the hepatocyte into the fluid that comes in through. This is sinusoidal type, so you got gaping holes between the within the endothelial cells that allow fluid to bathe the hepatocyte, but it's kept separate from the contents of the bile canaliculi. And here we can see This is the hepatic sinus right into there. There's a platelets going through. And then there's a space of these, and these are the endothelial cells, and this is the bile canaliculi and hepatocytes, as we could see. The hepatocytes here, there's this nucleus, and then there's cytoplasm there, is shown as glycogen. Stained for glycogen. You can see the kind of red structure is glycogen. This we can see is glycogen 2. It's not stained red, it's just a black and white image. But this is 2 hours after a fast, and you see 8% of the hepatocyte is glycogen. However, after a 24-hour fast, then the glycogen has been depleted mostly. And so this illustrates that the liver plays a role in the blood sugar concentration on a daily basis. And it makes up for if you don't have sugar intake from the digestive tract, it can make up that temporarily for during the day's time. Sugar and protein. Here we have sugar comes up, glucose comes in, stored as glycogen, and then is broken down, degraded to glucose again. And then also you got amino acids coming in. They make proteins, and then the proteins could be albumin, fibrinogen, prothrombin, lipoproteins that are produced there. And this is in the endocrine aspect of it, and that's separate from the exocrine aspect of it, where you have bile is being produced in the bile canaliculi. And here you can see where the two surfaces are separate. You have the sinuses, which are the endothelial cells, and the Kupffer cells lining them, and then you have the bile canaliculi, the yellow that you're seeing, that then gives rise to a bile duct. Now, the liver has different zonations. The classical lobule is the one I mentioned before that we see. There's also a portal lobule with a triad in the center, because the triad is a source of blood that goes to the central veins, and one triad could go to multiple lobes. Another way to look at it is the ascens, and that is the different layers. The first layer, if this is a triad and triad here, both of these can contribute to the central veins in both of these. And so you have the zone, the first zone, second zone, and third zone. So there's the classical lobule, portal lobule, and the ascens. And the ascens is the main one that's important. If you have liver damage due to a toxin, it will kill the ones in zone 1 first. And it would remove the toxin from it, so these cells are spared. However, if you have some kind of toxicant that will use up oxygen, these cells in zone 3 are already deprived of oxygen, so the toxin would kill these cells first. So if you have some animals that have died at a feedlot, and you look at the liver, and you'll be able to tell which cells were destroyed first, then it would tell you what kind of toxin you might be dealing with to help save the other animals in the herd. Here we see the liver. There's a central vein. You can see the kind of pink here are the hepatocytes. The solid kind of reddish color are the red blood cells that are in the sinuses. And here we can see many Kupffer cells throughout that has a role of cleaning the blood in the liver. Here we see another one. This is a gallbladder right in through there, but there's a liver here, a nice liver that we can see the triad, the 3 different components. You have the hepatic vein, hepatic artery, as well as the bile. And that's the triad components of it. And remember, the blood is going to the central vein, and the bile is coming back toward the triad. And here we can see the different cells there. We got the Kupffer cells, which cross over through there. Got endothelial cells, as well as hepatocytes. And the bile canaliculi, you can see the occluding junctions, the zonal occlutants and zonal adherents, which are preventing things from going between adjacent cells in the bile canaliculi. Here we can see the bile canaliculi right in through there. We can see it in through there, in through there. We can see the bile canaliculi between adjacent cells, and that's a different surface from that of the blood luminal surface that we see here and here, and also we can see some bile ducts as well. Now, if you stain a piece of liver for proteins that are in the bile canaliculi, this is what you see, that indeed the bile canaliculi touch all the hepatocytes and can be seen throughout. However, if you look at the bile duct is where you have concentration of the bile, but you don't have it distributed throughout like you do the bile canaliculi. The bile canaliculi has to touch each cell if the cell is going to contribute to the bile. Now, in the bile canaliculi, you have different things that are there. You got cholesterol in there, epidermal growth factor, insulin, and IgA. Body fluids have antibodies in it to help in the immune response. Also have bile salts and bilirubin that's located there too. So you have the bile canaliculi giving rise to the bile duct. There's a ductal on the bile duct, as you can see. So it giving rise from here to the bile duct, which has cube cells in through there. Then the bile goes through the common bile duct, and then it goes into the hepatic duct, you might say, a common bile duct, and then it goes through the cystic duct into the gallbladder itself, where it's stored. And here we can see the cystic duct, the hepatic bile duct, and this is the portal vein that is bringing the blood to there. And so here we can see in the common bile duct, you can see simple columnar epithelium, the same as what you see in the cystic duct, and also it's the same as what you see in the gallbladder itself. Now, in the cystic duct itself, you can see it's kind of closed there because you kind of have a swirling of the lining, the connective tissue, and the epithelial lining, as well as smooth muscle in through there, and that kind of prevents the collapse of the duct whenever you're changing the different pressures that might be involved there. So this is a cystic duct, which is the duct that goes to the gallbladder, and it has a little valve in there that regulates the flow of that. Here we see the gallbladder, some smooth muscle, a lot of connective tissue, and simple columnar epithelium covering the lamina propria. We can see the real specimen. This is liver out here, but then you can see simple columnar epithelium. In the lamina propria, there's lots of plasma cells associated with producing antibodies that would go into the bile that we mentioned before. You see how the enfoldings in through there increase the surface area, and also note that the cells can be separated out because actually it is pumping out water out of these. So it's pumping out electrolytes and pulling the water with them to concentrate the bile. Here we can see a higher magma, one of those. You can see the simple columnar epithelium that's there. We see a thick connective tissue layer. We also see a serous cavity with mesothelial cells on the surface that we can see right here and here, and connective tissue below, and more connective tissue when it has some smooth muscle in there as well. Now, one of the things you need to do with the GI tract is to be able to identify different cells on the surface. And here's the cardiac region. These are the three regions of the stomach. This is the fundic region, where you have surface mucus cells on the surface. You have protocells, chief cells in through there. Pyloric region, this is PAS staining. The intestine, simple columnar epithelium with goblet cells, and then here you can see the gallbladder, again, simple columnar epithelial cells, to be able to see the different ones. The mucosa that we want to look at is the surface epithelium, lamina propria, as well as the muscularis mucosa that's down below. If we switch horses to the salivary glands, and then we can see a salivary gland of a dog, and here we can see salivary glands of a human. The acessus is the functional unit of the salivary gland, and some of them are serous or proteinaceous secretion, some are mucus, and then some things are mixed. Salivary glands come from the ectoderm, the oral ectoderm, epithelial sheet, and then the GI tract comes from the endoderm, as you know. Now, saliva is important because it produces IgA antibodies. IgA antibodies are the secretory antibodies. So you have plasma cells produce antibodies, and you have transcytosis to transport that into the lumen. Transcytosis, as you know, transfer things from one surface to another surface. Also, it produces lactoferrin, which binds iron that's needed for bacteria to divide, so it prevents bacteria from dividing. Also, a lysosome that kills bacteria, and it constantly washes away dislodging bacteria as you swallow down the GI tract. And here we can see the salivary gland. It kind of has a host of different components to it. You can have a serous type, which has a nucleus in the middle, as opposed to the mucus type with the nucleus at the bottom. And this is a proteinaceous, more eosinophilic, and this is more lighter staining. Is the mucus. And then you also have these myoepithelial cells that surround it that squeezes it out. And if you go from there, you go to the intercalated duct, which is simple columnar epithelial cells. You may still have myeloid epithelium around it. And then you come to a striated duct. A unique duct is striated. You can see the lines in through there. And it's striated because you have infolding to the plasma membrane, and that lines up mitochondria. So the striations that we're seeing are really the mitochondria that are lined up because the plasma membrane has enfolded in through there, and that is the purpose of that is to increase the surface area to pump out the electrolytes. It pumps out the salts, leaving the liquid in through there to make a hypotonic solution. So you have the secretory portion goes to the intercalated duct, and from there it goes to the striated duct. So this is the intercalated duct. This is a mucus and a serous type secretion. Again, sometimes you can have the mucus cells surrounded by the serous cells, and that makes a serous demilume. So we call that a serous demilume that we threw there that we can see. You have intercalated ducts as well as the striated duct in the salivary gland. Here we can see the infolding of the plasma membrane and through here and there, and the lining up of the mitochondria, which causes the striations. And then here we can see the striations, which are really the mitochondria lined up through there. This is the secretory portion, and this is the striated duct portion. And I mentioned to you the purpose of the striated duct is to pump the sodium out to go to a hypotonic solution. Liquid is produced at 300 milliosmoles, but then by the time it gets into the interlobular duct, the sodium concentration has dropped down a lot, and through there osmolality has reduced, and that makes sense because if you're going to dilute foodstuffs, you need to have a hypotonic solution, not an isotonic solution. And so the striated ducts in the salivary glands pumps out the salts, so you have a more watery saliva. And here we can see the salivary glands. This is the secretory portion that you see, the intercalated duct, kind of cuboidal cells lining through there. You can see it goes from the intercalated duct. Intercalated duct goes to the striated duct. Here we can see a striated duct here, and you can see the mitochondria have lined up there. The infolding of the plasma membrane lining up on the mitochondria has made these striated ducts visible. Salivary glands is a compound tubular acinar gland. So these are These are acini, but also have a tubular component to them as well. You can see a tubular component right there. There's the mucus and the serous, and this would be a serous demilune in the case of here. So you have a secretory portion of the acinus, then you have the intercalated duct, striated duct, and then some kind of excretory duct. Serous, mucus, and the myoid epithelial cells. You can see the myoid epithelial cells that contract to squeeze it out. And here we can see it's going from a mucus type cell to the intercalated duct, from the intercalated duct to the striated duct, that you can see the process of going from one to another duct, as you can see. We can see again, these are striated ducts in through there. You can see the a little bit of striations associated with them. There's also intercalated duct that we can see in through there. There's another intercalated duct, another striated duct. Also, we can see some fat cells distributed out through there. There's a vein, there's an artery. Another one, we can see the serous and the mucus secretions. Again, the serous has a spherical nucleus, and mucus is off the base. The mucus has a flattened nucleus and a kind of lighter staining cytoplasm, as you can see here. This is a serous, this is the mucus, and here we see the striated ducts with the lining up by the mitochondria. And this particular one, we can see a nerve in through there, as well as mucus and serous secretions that are there. The pancreas is the last organ, and here we can see some ducts in the pancreas. You have the islets of Langerhans, you have the pancreatic acinar cells, and these acinar cells empty into an intercalated duct, and then that empties into an intralobular duct, but the intralobular duct is not striated. There's no striated duct in the pancreas, but the intralobular duct connects the intercalated duct with the interlobular duct, but it's not striated in the pancreas. So in the pancreas we have both exocrine with the pancreatic acinar cells and their duct system, and the endocrine with the insulin and glucagon being produced in the islets of Langerhans. So we'll see how things are organized. They're organized and so that the endocrine part is distributed throughout the pancreas. And we can see that the islets are there. We'll be able to see the ducts and the acini and how it works. So if we look at a piece of pancreas, we see the pancreatic acinar cells and acinus here. We can see some connective tissue, and we see these lighter areas, which are the pancreatic islets or the islets of Langerhans. You can see them, they're distributed throughout the pancreas. They're distributed in the pancreas because they have a local portal system between the islets and the pancreatic acinar cells. And that's why it needs to be distributed throughout, because there's a local portal system around each one of these islets. Regarding the duct system, of course you have the acinar cells are serous type with zymogen granules near the lumen, which gives a red color to it. The blue tint in the bottom is the ribosomes of the rough endoplasmic reticulum associated with making those proteins. And from the acinus, you have these central acinar cells, which is one thing is pointed out here, and that's actually the beginning of a duct. And that beginning of the duct is the intercalated duct. And then the intercalated duct goes to an intralobular duct, and then an interlobular duct. But the intralobular duct, not shown here, is non-striated. Indicates the pancreas because everything is isotonic. You're not producing a hypotonic saliva in the case of the pancreas. So here we can see the pancreas, the intercalated duct. It looks very similar to what we saw in the salivary gland. You see it here. You see one here, and you see one coming there. You can also see the central acinar cells. So you can see these cells here actually, they're inside an acinus, but they're starting to duct. And here we can see an acinus there, the cells starting a duct that continues continues there. And so here we can see the central acinar cells inside the acids, and then this is an intercalated duct that appears there. Another view, you can see the intercalated duct, again, like simple cuboidal epithelium surrounding the thing. And then we see the clear islets of Langerhans, and we see that they're distributed throughout the pancreas. And this is where you have the alpha and beta cells. The beta cells are more numerous, and they're more in the center of the islets. And then we have major blood vessels in the connective tissue, nerves, of course, in the connective tissue. And here's the interlobular duct. So you go from an intercalated duct to intralobular to interlobular, but the intralobular is not striated in the case of the pancreas. And here we see the pancreatic acinar cells with lots of rough endoplasmic reticulum, a big nucleus, euchromatic nucleus with a big nucleolus associated with making a ribosome on RNA, and then finally the production of zymogen granules. You see some ones that are less condensed, and finally they're released at the surface. And here we can see the tight junctions between adjacent pancreatic acinar cells, and also between this acinar cell and this duct cell. So this is the starting of a duct. So this is part of a duct, the intercalated duct that we can see. And here we can see the islets of Langerhans. Here we can see the pancreatic acinar cells and the acids. We can see it here with the zymogen granules located next to the lumen where it's discharged. You can see the central acinar cells right in through there, which is actually the beginning. And you see this multiple times. That's actually the beginning of a duct. And here's the intercalated duct, another intercalated duct. And these guys here are the interlobular ducts that connect, or in the connective tissue there, that are between the different lobes of there. So in summary, the digestive tract associated organs do important things. They help the defense against pathogens. They store and release nutrients on demand, and they also help to produce digestive juices and help in antimicrobial growth of things. We have some questions on the liver, pancreas, and salivary glands. The humoral activity of the immune system is illustrated by the transfer of IgA, immunoglobulin, by the epithelial cells in which of the following fluids? Saliva, yes. Milk, yes. Bile, yes. The answer is E. All these body fluids will have antibodies associated with them. Which functions do not match the gallbladder? Which functions do the gallbladder and the urinary bladder have in common? Temporary storage of waste products? Yeah, bile and urine. Concentration of respective luminal contents? No. The urine does not concentrate it. It is concentrated in the gallbladder. The secretions are. And then the bile is, I should say. Also, you got similar type of aluminum? No, they're not similar. You got transitional in the urinary bladder, and you got simple columnar in the gallbladder. Characteristics of the pancreas include a portal blood vascular system. Yes, between the islet cells and the pancreatic acinar cells, there's a portal system. Also, the endocrine cells are in the islets of Langerhans. Yes. The acinar cells and striated duct? No, there's no striated duct in the pancreas. Striated duct is the function of that is in the salivary gland to make a hypotonic solution. You don't need a hypotonic solution in the pancreas. Everything is isotonic, so the answer is D. We want to thank the books for which you made some drawings that we chose and a couple images I showed from Body Worlds. We thank those for their contributions to the process. When I went to Colorado in 1973 or so, we went on Christmas time. We went to see the mountains up top, and This is a little town of Silverton. We went there and got there at night. I was so happy to see Silverton. That's the end of Medical School Histology Basics, liver, gallbladder, salivary glands, and pancreas. Thank you.